Okay, so I think that we can start. It is my pleasure to have Sebastian Schmidt here to start the DSB2021 workshop with a talk titled Genome Assembly, Universal Theoretical Framework, Unifying and Generalizing the Safe and Complete Algorithms. Thank you again, Sebastian. The stage is yours.
Thank you very much. It's also a pleasure for me to talk here today. So, first of all, the project you already heard. My co-authors on this project are Massimo Cairo, Shahbaz, Romeo, Rizzi, Alexandru, Tomescu, and Elia Zirondelli. And today, I will first of all give you a short introduction about genome assembly and safety in genome assembly, and then also motivate why we might want more, in practice, more intricate models of genome assembly. And then I will introduce to you the current safe and complete algorithms in genome assembly. These are only for rather simple models of genome assembly. Then I will introduce to you the hydrostructure, which is a theoretical construct that is able to aid us in all the more complicated models of genome assembly for the characterizations of safe solutions as well as the algorithms. And then I will introduce to you how the algorithms work a little bit. So first of all, genome assembly and safety. So in genome assembly, we usually have a genome graph that's used by almost all modern assemblers. And in this graph, each edge is part of the original genome, and the genome is a walk in the graph. And for example, if we know that the graph is built from a single circular genome, then in this graph, our original genome would be a single circular walk. On the other hand, if we would know that there are multiple circular genomes in our data, then we would have multiple circular walks that represent these genomes or chromosomes, for example. Now, you might notice a challenge here. It's not unique. What is the correct solution walk in this graph? So what do we do in practice about that? We find only safe subwalks of the true genome. These are those walks that are subwalks of each possible solution walk. So they are then definitely part of the original genome. And now that we only report subsolutions, we want to be as complete as possible because we want to report as large solutions as possible still. Okay, so much for the short introduction. Now let's come to the practical issues. So let's look at some genome, for example. I have here a genome from a diploid species. There's a copy from the father, a copy from the mother, and this one's linear. And here there's a single nucleotide polymorphism, SNP. Let's construct the genome graph of this genome. This is an edge-centric de Bruijn graph, and it would look like this. And I drew here as a blue line the genome, the copy of the mother, the chromosome of the mother. And now we've seen this graph, for example, over here. The structure here is called a bubble. It happens when there's, for example, a slip in the data or just some error in the data that we would need to resolve now. And then also in practice, we often get some edges, I call these red here, where the coverage is low. And then it's not always clear to say if these are actually part of some of the genomes or if these are actually errors. And let's look at like how a classical assembler would handle these, this kind of genome graph. So first of all, it would pop this bubble. It would get rid of all the low abundance edges along with their nodes, and then it would compute unit ticks on this graph. Unit ticks are paths where the inner nodes have in and out degree 1. So like this one here, for example. And as you can see, if we assemble the unit ticks of this graph, we get down here, I aligned the unit ticks to the genome, we get a fragmented and also assembly that also misses some parts. On the other hand, in this example, if we would now apply a more complete algorithm for a more realistic model. So this model, we now assume that we make these red edges here not covering. That means they do not need to be covered by the solution walks. They just can be covered if the rest of the graph topology requires it somehow. And also here for this bubble now, instead of popping it, we keep it here, but we mark it as invisible. And then we can later on, like this hides it from the safe solution walks from the context. but we can later on come back and resolve it with some other methods. And if we now compute the safe and complete solution of this genome graph, then we get these green contexts down here. You see there's less of them, they're less fragmented, and they also cover the whole genome much better. So this is the motivation for why we would want more complicated or more intricate models of genome assembly in practice. And let's quickly look at what currently exists in terms of safe and complete algorithms. So for the single circular model that we have seen in the beginning, there are omnitics. This is the characterization of the safe walks in this model. They were characterized in 2017, and there was an optimal order of MN algorithm that was published 2019, and then also an output optimal algorithm. This runs in size linear to the input, plus size linear, runtime linear to the size of the input, plus runtime linear in the size of the output. And this was published in 2020. And this is actually useful in practice because while in the worst case, the total length of all the omnitics is order of MN, in practice is usually much lower. And there's also an algorithm for the multi-circular model that runs in order of M squared N. And these algorithms are all based on theory of forbidden paths. What's a forbidden path? Well, if we look at this blue walk here, a forbidden path in this blue walk would be a path like the red path here that repeats an inner subwalk of the blue walk. And if such a path exists, then this blue walk would not be an omnitic. If no such path exists, then this blue walk would be an omnitic. Now, this is how these older characterizations work. Now we also want to, like as I motivated in the beginning, solve more advanced models. For example, if we know that we have two copies for each chromosome, we want to have maybe a fixed number of solution walks. We might want to model linear genomes. Most vertebrate species have linear genomes. And we, as I motivated in the beginning as well, we might also want to model that only a part of the graph needs to be covered, as well as we might want to make part of the graph invisible. And now we would not want to, again, go for each of these models, for each of the combinations, through the whole theory again. But instead, we would like to have one unified theory for all possible combinations of these models. And for this, we introduce the hydrostructure. The hydrostructure is defined on a walk W, and it's defined in a strongly connected graph. For not strongly connected graphs, we can use the hydrostructure in the strongly connected components, and then do something else in the edges between these strongly connected components. But the hydrostructure actually solves the part that's hard to solve. The part between these strongly connected components is simpler to solve. And for the purpose of this talk, I will restrict myself to W being, first of all, a path, so it doesn't repeat any nodes or edges. And then also, I want it to be a non-trivial hard. What is non-trivial hard? A non-trivial hard is a walk that starts with a join arc. Usually this is a join arc because there are two incoming arcs into this node here. And it ends with a split arc. This is a split arc because there are two outgoing arcs from this node here. So we only consider these kinds of walks. And now I will first of all describe the hydrostructure from a bit more abstract level. Then we will go into the definition. We will look at some important characteristic of the hydrostructure. And then I will explain to you how it is applied in practice. So first of all, the hydrostructure decomposes our graph into four regions: the cloud, the vapor, the sea, and the river. And there's a restricted connectivity between these regions. This is motivated a bit by the water cycle. This is where the naming comes from. So for example, from the cloud, water rains into rivers or dissipates into vapor. From the river, the water flows into the sea. From the sea, water evaporates and then collects afterwards as clouds again. This is how the connectivities work in the structure. And now in the structure, we have a very important property. That is, if this vapor here is a path, then our walk W is a C-cloud bottleneck. What does C-cloud bottleneck mean? C-cloud bottleneck means that whenever we go from C to cloud, we need to use the whole W as a subwalk. And this is now very useful for our safety problems, because now for any model of genome assembly, if we can prove that each solution walk in this graph goes from C to cloud at least once, then we have proven that our walk W is safe. On the other hand, if we have a counterexample, if there is some solution walk that does not go from C to cloud, then we have a counterexample, and then we know that W is not safe. Okay, so much for the abstract overview. Let us go into the definition. So I have again the walk W. I call the first edge F, the last edge G. And now we define two sets, R plus of W, R minus of W. R plus of W is everything reachable from F without using G. So it's this colored region down here, the vapor and the C. R minus of W is everything backwards reachable from G without using F. So it's this colored region up here, the cloud and the vapor. And now we define our four sets. The cloud is R minus without R plus. The C is R plus without R minus. The vapor is R plus and R minus, the cut set. And the river is everything else. And now, just by this definition, we get our property that our W is a C-cloud bottleneck if and only if the vapor is a path. And since this property is so central, let's also look at the proof idea of this property. So I brought here again the definitions of R plus of W, the definition of R minus of W, and here a definition of the C-cloud bottleneck. So W is a C-cloud bottleneck if each walk from the C to cloud has W as a subwalk. And now we can look at our lemma. So the bottleneck property is that W is a C-cloud bottleneck if and only if the vapor is a path. And to prove this, we first of all notice that our arc F is the only way to enter R minus, because R minus searches backwards from G and only stops at F, searches in G without F. Symmetrically to that, our arc G is the only way to exit R plus, because in R plus we search forwards from F and we only stop if we reach G. And this gives us a nice subproperty. That is that each walk from C to cloud, when it needs to, first of all, enter R minus at some point, it needs to exit R plus at some point, and therefore it needs to use F and then G. Therefore, it also uses the head of F and the tail of G. So it has some head of F, tail of G subwalk that we call X now. And if we now take a minimal one, then we know that this one is in the vapor, because then it doesn't repeat the head of F or the tail of G, and our walk W here is always in the vapor. Now we can assume that W is a C-cloud bottleneck, and in this case we know, okay, each walk from C to cloud has to use W. So then the only way for this walk X to be, since it's like starts from the head of F and the tail of goes to the tail of G, is must be W. Otherwise, there would be a different walk from C to cloud that doesn't use W. So therefore, since X is in the vapor, we know that our vapor now can only be one choice. That's the path. And on the other hand, if we know that the vapor is a path, then we know our walk X goes through the vapor. So there's only one choice for our walk X. And therefore, since like this is each walk from C to cloud, we know that also then each walk from C to cloud has only this one choice. Therefore, W is our C-cloud bottleneck. Okay, this is the bottleneck property, and this is what the core of the hydrostructure, so to say. Let us look at how we apply the hydrostructure in practice. So, as I said before already, for any model, if we can show that each solution walk goes from C to cloud, then we know that our walk W is safe. And this now simplifies already the single circular model, because we know that if there's an edge in the C, an edge in the cloud, then definitely we need to go from C to cloud once to connect them both with a single circular walk. On the other hand, for multicircular, if there would be no river now, so we imagine the river to be gone, no red parts here, and we have an arbitrary amount of multicircular walks now, so we can have one circular walk that covers the cloud and the vapor together. We can have one that covers the cloud and the vapor and the C together. And then we have covered the whole graph. Remember, the river isn't there without ever going from C to cloud. So we have a counterexample. Our walk would not be safe. On the other hand, if there is a river, we remember from the connectivities, the river can only be entered from the cloud, can only be exited into the sea. So to cover the river with a circular walk, we definitely need to go from cloud to sea, and then also from sea back to cloud, because our walk is circular. So if there's a river, then in the multi-circular model, our walk W is safe. And we can iterate the same for the single and multilinear models. We can iterate the same for the subset covering models. We can iterate the same for subset visibility and for all the combinations of these models. And we always get to some useful characterization. So this is how we characterize our walks using the hydrostructure, our safe walks. Let us now look at how we compute the safe walks, how the algorithms work. So first of all, simple algorithm, the verification. So we are given a walk W again, and we ask is this walk safe or not for a given model. In this case, we can construct the hydrostructure in order of M time. So M is the amount of edges. And then we can, in most of the models, verify also in order of M on top of the hydrostructure the characterization. So in these cases, we get order of M verification algorithms. In some cases, when we have a linear model with a certain number of chromosomes, or if we have subset visibility, we get order of Mn here. On the other hand, for enumeration algorithm, there's a useful property of the hydrostructure. That is that we can construct it incrementally. That means we can construct it for all subwalks of a given bottleneck walk at once. And this works because if we imagine this whole walk here to be our bottleneck walk and the highlighted walk here to be our subwalk, then if we now shift this subwalk to the right along the bottleneck walk, then the sets R plus, the set R plus always increases, the set R minus always decreases. So the C always increases, the cloud always decreases. And this allows us to amortize the costs of the hydrostructure computations for the different subwalks into just order of M, so into just one hydrostructure computation. And then we can use this to enumerate the safe subwalks for a single given bottleneck walk in order of M plus size of the output time. And here again, we have an exception for some linear models and for subset visibility. But now we want to, of course, compute all the maximal safe walks in the graph and not just the subwalks from a single bottleneck walk. And for this one, there's another nice property, that is that the safe walks in most of the models are subwalks of omniticks. And we know that omnitic enumeration takes order of Mn time. And also there's another property that actually only order of N of the omniticks are bottlenecks. And so only for this order of N, we need to build the hydrostructure and do this more complicated algorithm to find the safe subwalks. For the not bottleneck walks, actually, the conditions it's very easy to verify safety or unsafety. It works in order of the length of the walk otherwise. So then we get in total a runtime of order of Mn plus this order of N times order of M plus O. So we get an order of Mn plus O, where O is again the size of the output. To enumerate all the maximal safe walks in any given model, except now here only for linear with a certain number of chromosomes, we get order of M squared times N. And this already concludes it. So we have now seen that with the hydrostructure, we can unify the theory of safe and complete genome assembly and also solve more advanced models, as well as get optimal algorithms for most of our model combinations, except for one model combinations, as you have seen. So we can apply it nicely. Here are my references. And then thank you very much for attending. And if you have questions, I'm happy to answer.
So please, are there any questions? Because I cannot see any questions in the chat or in the list of questions. All right, I have one curiosity. So you have clearly you clearly said that it is a theoretical framework. But do you think that the algorithms that you have described can actually be implemented, not by you actually? So I mean, or are they your theoretical interest only?
So actually, the basic omnitic algorithm on perfect data, so on error-free reads, gives an improvement of 60% in terms of contact length and biological content. So this actually is a very significant improvement in practice. These algorithms here, I think they also can be implemented, and they probably work better hand in hand with graphs or assembly graphs that have some errors or some uncertainties, which is much more common in practice. So like, I think we can actually get some improvements in some assemblers using these kinds of algorithms.
Thanks. So I can see now one question in the chat. Does this framework lead to longer safe strings than omnitics?
That's an interesting question, actually, because first of all, it leads to shorter strings because we have all these strings as subblocks of omnitics. But on the other hand, now we can like take a genome graph with more uncertainties. So we don't need to delete uncertain parts of the genome graph. So we hope that in the end, we might even get longer strings using this.
Okay. Another question is, does the repeat graph, as used in the fly assembler, fit in this model?
This is also a nice question because I actually asked it to myself before, but I couldn't answer the question yet. So I would hope that it does. If it's like a genome graph and it's reasonably accurate, then I'm pretty sure that it also fits into these models, but I didn't try it out yet.
And does this model rely on read length? So can it be applied to both short and long reads?
It can technically be applied to anything where you can build a genome graph from. So if you can build from your reads a graph in which the original genome is a walk, then you can apply these models. So also from long reads. There's, for example, the assembler Watchtower, which builds a genome graph, and like this fly assembler also has a some kind of genome graph. Then we can apply this in these cases as well. Like that.
Okay, thank you very much. Thank you very much. So chat. Okay, yes, and let's see if there are any more questions, but no more questions. So thank you again. A beautiful start to the workshop.